So I'd like to thank Anne and the Academy for the invitation to speak, and you all for the time to listen to this talk. In the next 15 minutes, I'm going to use examples from my group and associated literature to share why I'm interested in understanding molecular modes of action of potential drug molecules to their protein targets, and illustrate why this knowledge can be helpful in finding new medicines. So we all recognise that choosing the right therapeutic targets to work on is really important in drug discovery, but also very difficult. These papers and others have suggested that choosing targets with genetic disease associations and using advanced technologies such as computational approaches may enhance our chances of success. Unfortunately, target associations by themselves are not sufficient to tell us how to design the exact molecule that can restore normal health and function. So even if we choose the right target we'll work on, what we really also need to understand is the molecular mode of action required for an efficacious and safe drug. To answer questions such as, what part of the protein molecule does a drug need to bind to? Does this binding need to inhibit or activate protein function? And if there's more than one function, is there a relevant function we want to perturb and one that we want to leave well alone? Does persistency of binding matter for efficacy? And is there a margin of specificity over other protein molecules a molecule has to have to be safe? The reason why these molecular questions are important is because what we tend to do in drug discovery is change the atoms within our drug molecule. But of course, what we want to do is affect human pharmacology. So knowledge of that molecular mode of action is really to do with understanding the relationship between those things that we can alter, the atoms in the molecule we make, the target we are aiming for, the mode of engagement in the two, and what we'd like to alter, which is the pharmacology. So early insight into that optimal molecular mode of action allows us to identify which of the many attributes we could change, which is the most critical for a given target in a specific indication. And this can help us focus our search for new medicines and therefore help us to get us there faster. So my group is focused on the very early stages of drug discovery, and we use a combination of structural and biophysical methods to gather information to unravel molecular modes of interaction. To answer specific questions such as, does a compound bind to one or maybe a multitude of targets? What are the molecular details of that interaction? And how do these structural elements correlate with function and pharmacology? So today I'm going to be highlighting two examples on elucidating and in particular visualizing the molecular mode of action. Firstly, in opening up a previously unexploited class of epigenetic proteins to drug discovery, the bromodomains, and secondly, in informing opportunities to counter antibiotic resistance when targeting topoisomerase enzymes. In humans, we have about 1.6 meters of DNA that has to be condensed into the cell nucleus that's only about 20 microns. DNA is tightly coiled and wound around histone octamers to form nucleosomes, and then further compacted into chromatin. For any gene to be transcribed, that DNA has to be made more accessible. And epigenetic post-translational modifications can do this as they alter chromatin structure. Some of these modifications have a direct impact on the physical properties of nucleosomes, but a more general mechanism by which these modifications have an effect is by recruitment to the site of modification specific reader domains, such as the helical bromodomains shown on the bottom right here, which are a family of about just over 30 proteins that recognize acetylated lysines, which on histones are a signature for gene activation. Now, it turns out that post-translational modifications rarely occur in isolation. What you get is a complex pattern of modifications, sometimes known as the histone code. Similarly, reader domains rarely occur in isolation. They can be concatenated together within the same protein to read these patterns of modifications, or brought together by virtue of the large protein-protein complexes that assemble on chromatin. In the middle is the 4 helical bundle typical of a bromodomain reader. Single or multiple bromodomains can be found on a protein to sense acetylation patterns leading to downstream additional recruitment of proteins to the site. On the right shows branch 2 of the BET family of bromodomains, consisting of 4 members, BRDs 2, 3, and T. Each of these proteins have 2 bromodomains, the pale BD1 and the dark blue BD2. Given, I hope, what I've illustrated is the complexity of bromodomain histone interactions, finding compounds that directly disrupt these interactions and liberate bromodomains from chromatin was found to be very difficult, if not intractable. Certainly no known compounds were known to do this until we and others stumbled upon them largely by accident. In our case, what we were doing was looking for ApoA1 upregulators for dyslipidemia in a luciferase reporter assay in HepG2 cells. We didn't know what target we would find or compounds, and in fact, whether we would find any at all. But compounds we did find. In fact, several series, including the benzodiazepine series you can see on the left here, which we were able to deconvolute using a mixture of chemoproteomics and biophysics to be rather unexpectedly selective binders of all 8 bromodomain modules of the BET proteins, all BD1s and all BD2s of these proteins, whilst remarkably sparing the rest of the bromodomain family tree. We were also successful in visualizing how these compounds bound using X-ray crystallography. The acetylated lysine of the histone peptide, shown in yellow, binds deep in a pocket that's conserved amongst all bromodomain families, whilst the context of that peptide is recognized by the flanking orange BC and longer ZA loops at the top of the 4 helical bundle. This variability is what allows different bromodomains to recognize different acetylated protein partners. Our inhibitors, they bind in the same site as histone peptide. They're therefore direct orthosteric antagonists of histone binding. What we quickly recognized from our many structures was that nature's modular approach with modified histone peptides could be used to make selective inhibitors outside of the BET family by designing a genetic library of chemical anchors that mimic acetylated lysine, and then building these out to exploit the loop regions that are different amongst family members. And this is what I'm going to show you. So this side view slices through the acetylated lysine pocket of BET. In green is IBET762. This is a molecule we've progressed into clinical trials for hematological malignancies, solid tumours, and midline carcinoma. Some of the key interactions needed to mimic acetylated lysine in the pocket are shown in grey. These are 3 examples of how very different looking compounds can all act as acetylated lysine mimetics by preserving the grey interactions. There are 3 from a library of 700 possibilities that could be used as anchor points to start our search for bromodomains targeting members inhibitors, targeting members beyond the BET family. This is because if you start looking beyond the conserved pocket, as shown here for CREB and BET bromodomains, a single amino acid difference to the left of the pocket suggests that bulking up this yellow starting point on the left-hand side of the molecule would be tolerated in CREB, but in BET would quickly bump into the protein. What this means is we would have a compound that's no longer able to bind BET and therefore selective for CREB, which is why, far from being an intractable target class, it's been possible for us and many groups to exploit this knowledge from unraveling the mode of action of these pan-BET compounds to optimize molecules across the broader bromodomain family. These insights also offered the possibility of designing new pan-BET inhibitors that was done by a large number of other groups as well, which has led to several of these compounds entering clinical trials with over 20 registered trials, largely for oncology indications. Ironically, the strong and broad pharmacology that enabled us to find these pan-BET inhibitors in the first place may ultimately limit their tolerability. But what we now know about these mode of actions has put us in a great place to consider ways of narrowing down their breadth of pharmacology. And what we've been able to do is design compounds that exploit the protein differences between the first and second bromodomains to give domain selective inhibitors, as it turns out that BD1 and BD2 domains may have distinct roles in proliferation and inflammation. Make conjugated compounds with targeted motifs, such as the esterase-sensitive motif, and this results in higher exposure in a subset of cells, in particular myeloid cells, that may make these compounds more suitable for inflammatory indications. And we've also been able to demonstrate using PROTAC technology to create compounds that have a resulting proteasomal degradation of the BET proteins in a subset of cells that depend on the presence of a specific E3 ligase rather than just uniformly inhibiting all cells. So in summary, understanding the molecular mode of action of pan-BET compounds has opened up inhibitor design for other bromodomain modules across the family and given us opportunities to try and maximize our chances of getting BET compounds to medicine by designing BET modulators with a variety now of new molecular modes of action. And this may provide new options to find the appropriate balance of efficacy and safety for different indications. My second example focuses on topoisomerase antibiotics. Unlike bromodomains, many proteins need to change shape to carry out their normal function. An example is topoisomerases, which are the molecular targets of fluoroquinolone antibiotics, such as moxifloxacin. Being able to characterize and visualize this dynamic reaction gives opportunities to target a variety of conformational states that may lead to different biological effects, in this case in our search for new antibiotics that counter resistance. Currently, there are 2 clinical phase 3 topoisomerase inhibitors for priority pathogens that the WHO has deemed innovative. The first is gepotidacin, the other is zoliflodacin. Topoisomerases have to unwind compacted bacterial supercoiled DNA to allow transcription to occur. This is heavily dependent on conformational change. Binding of the DNA duplex across the symmetric topo enzyme causes both changes in the protein and the DNA, with the bound DNA stretched across the 2 symmetric catalytic sites. Then in pretty much the same way that you might take a shortcut in trying to untangle a ball of string if you had access to a pair of scissors, the bound strand, each strand of the bound DNA is first cleaved to give single and then double-stranded breaks. A second duplex of DNA is passed through the gap created. This exits and the first strand is religated. So inhibitors of this enzyme could prevent DNA binding in the same way that the BET inhibitors directly prevented histone recognition. But it turns out for this enzyme, it's much easier to find compounds that stabilize one or more of these intermediate states. So either prevent the conformational change, prevent the cleavage, or its religation. The result is an enzyme that's trapped in a stored non-productive state that prevents replication. Additionally, you get accumulation of unpaired DNA breaks, which can in itself induce cell death. Our structural studies using the core Staph aureus topo2 enzyme has allowed us to visualize some of these stored complexes and to identify at least 3 inhibitor sites near the DNA gate. So this is a composite image where the protein and DNA are taken from an X-ray complex with moxifloxacin, and the other inhibitors have just been overlaid onto that structure. Site 1 is the catalytic site. This is where moxifloxacin binds symmetrically with 1 molecule bound at each active site. It sits deep in the DNA gap caused by cleavage. Its mode of action is to just sterically block religation. QPT1 and its derivative, zoliflodacin, also binds at the same site and has the same preference for doubly cleaved DNA. Gepotidacin, an allosteric inhibitor, binds in site 2 at the dimer axis of the complex. Half of the gepotidacin is wedged between the central base pairs of DNA, the other half in a protein pocket at the dimer interface. Its preference is singly cleaved DNA complexes, so it gives a different distribution of aborted DNA products to site 1 binders. Site 3 is another allosteric site, where we have found a distinct series of thiophene. This is a hinged site, surrounded only by protein and far away from DNA. The mode of inhibition is to restrict protein movement necessary to transition between DNA binding, cleavage, and religation, which means it's indifferent to the DNA state. Hopefully you can see that binding at these 3 sites may lead to different resistance profiles and why it might be beneficial to have access to molecules that access each of these different molecular modes of action. This slide shows the full X-ray structure of the stored DNA protein inhibitor complexes, illustrating the conformational preferences in both the DNA and protein more clearly. So as you go from double, single, and then separated DNA being bound. So in summary, in this very dynamic system, visualizing the conformational dynamics in trapped states has rationalized the differential consequences of inhibition and resistance profiles. It's allowed us to understand how to optimize, combine, and design new compounds to these stored states. Furthermore, I think understanding the complex conformational landscape may highlight yet unexploited opportunities for intervention. So I'd like to thank you for joining me in this whistle-stop tour of molecular mode of action. I hope this has illustrated the benefit of being able to unravel and particularly visualize this, both in fully exploiting known systems and opening up opportunities for new targets. I'd like to end with acknowledgements of the many, many people who've contributed to this work within GSK and our collaborators. Really too many to list individually, but I'd like to particularly single out my personal thanks to Paul, Manu and Rav, with whom I've worked many years on bromodomains, Ben Bax, who now in Cardiff, who was a key driver of the structural topoisomerase work. And finally, I'd like to extend my congratulations to all of the 2022 elected fellows, in particular Imre Georges and Mike Blackman, whose own scientific contributions have enabled, informed and inspired similar structural and molecular mode of action work in other areas.